Let's start with chemiosmotic hypothesis now. So this chemiosmotic hypothesis is given by the scientist Michel. Hence it is also called as Michel's hypothesis. Okay. So he gave this hypothesis, chemiosmotic hypothesis, to explain the formation of ATP during photophosphorylation that occurs inside the chloroplast and during oxidative phosphorylation which occurs inside the mitochondrion. Okay, so he gave this hypothesis to explain the formation of ATP, which occurs during photosynthesis that occurs inside the chloroplast and oxidative phosphorylation that occurs inside the mitochondrion. So this occurs inside the mitochondrion, whereas photophosphorylation occurs inside the chloroplast. So formation of ATP inside the chloroplast and even inside the mitochondrion is linked with the development of proton gradient. Now let's see where this proton gradient is developed in chloroplast and where this proton gradient is developed in mitochondrion also, and how this proton gradient is developed. What are the reasons, what are the factors for the development of proton gradient inside the chloroplast and even in mitochondrion. Okay, so what happens inside the chloroplast during electron transport? So during electron transport, that is during electron transport system, as the electrons are transported through carriers, there is pumping of protons. When the electrons are transported from one carrier to another carrier, there is pumping of protons. Protons from where? From stroma to lumen. So there is pumping of protons from stroma to lumen. So inside the lumen, proton concentration increases. That increase in proton concentration is called as development of proton gradient, which is nothing but proton motive force. Okay, so due to the increase in proton concentration inside the lumen there is development of proton gradient, which is nothing but proton motive force is developed. Now this proton motive force which is developed inside the lumen is responsible for the formation of ATP. It is responsible for the formation of ATP. Now let's see where this proton motive force is developed inside the mitochondrion. Right. Inside the mitochondrion also ETS occurs, electron transport system occurs. Where it occurs? It occurs on the surface of inner mitochondrial membrane. So when the electron transport occurs in the inner mitochondrial membrane, that means when electrons are passed from one carrier to the another carrier present in inner mitochondrial membrane, proton pumping occurs. From where? So protons are pumped. Proton pumping occurs from matrix to intermembrane space. From matrix to intermembrane space of the mitochondrion. Intermembrane space. I am writing it as IMS, intermembrane space. So what happens inside this intermembrane space? Concentration of protons is increased. So due to the increase in concentration of protons, proton gradient is developed, that is nothing but proton motive force, which is responsible for the formation of ATP. So proton motive force is developed, which is again responsible for the formation of ATP. Okay, now let's study how this proton motive force is developed inside the lumen of the chloroplast. Okay, so this is thylakoid membrane. Let's say this is thylakoid membrane. Inside this membrane, what is present? Photosystems are present, that is photosystem 2 and photosystem 1. We already studied in previous videos about these photosystems and about electron transport, that is non-cyclic and both cyclic electron transport system also. So this is thylakoid membrane. Thylakoid membrane. Thylakoid membrane. This is lumen. This part is lumen. This is lumen part, and this part, outer space, which is covered by outer space of this thylakoid membrane, which is covered by inner membrane of chloroplast, is stroma. So this part is stroma. Right. So in this thylakoid membrane, what is present? Photosystems are present. Pigments are present in the form of photosystems. So this is photosystem 2, PS2, and this is photosystem 1, PS1, and here I am drawing the cytochrome BF complex. So this is cytochrome BF complex, right. So to this photosystem 2, water molecule is associated transiently towards the inner side of the thylakoid membrane, that is towards lumen side. Water molecule is transiently associated. So when the reaction center of this photosystem 2 absorbs sunlight, what happens? Electron gets excited. Reaction center of the photosystem 2 gets activated at the absorption when it is absorbing 680 nanometers wavelength of sunlight. So it shows absorption maxima at 680 nanometers wavelength of sunlight, and hence electron gets excited. So when the electron gets excited, what happens? The reaction center of photosystem 2 becomes a strong oxidant. As it becomes a strong oxidant, now it causes the oxidation of water molecule, which is transiently associated with photosystem 2, that is towards the inner side of the thylakoid membrane or lumen. So this water molecule undergoes splitting into 2 protons, 2 electrons, and 1 nascent oxygen. Okay, so this nascent oxygen and protons are released into lumen, whereas electrons enter into the reaction center of photosystem 2. From here, these electrons are transported through electron transport system, that is through one carrier to the another carrier present in the electron transport system of this thylakoid membrane. So first electrons are passed on to pheophytin, which is attached to the outer side of the thylakoid membrane. From pheophytin, now electrons are transferred to PQ. PQ is nothing but, it is a proton carrier which is present towards the inner side of the thylakoid membrane. So this PQ is originally a proton carrier, it is not an electron carrier. So when it receives 2 electrons, it consumes 2 protons from, per 1 electron it consumes 1 proton from stroma. So per 2 electrons it consumes 2 protons from stroma, and leaves these 2 protons into lumen, and passing on electrons to the next carrier, that is cytochrome BF complex. From cytochrome BF complex, now electrons are passed on to the next carrier, that is plastocyanin. From plastocyanin they are passed on to PS1. So now electrons have reached PS1. And the reaction center of this PS1 also absorbs light energy, and the electrons present in PS1 also get excited and they enter into FES. FES, that is the initial electron acceptor of photosystem 1. Initial electron acceptor of photosystem 2 is pheophytin. From FES, these electrons are transferred to FD, that is ferredoxin. From ferredoxin, now electrons are picked up by NADP+. So that NADP+, after receiving 2 electrons, it consumes 2 protons from stroma, and it is reduced into NADPH plus H+. So this reduction of NADP+ into NADPH is catalyzed by an enzyme NADP reductase. I am writing the name of this enzyme here, that is NADP reductase. Okay. So due to the pumping of protons, due to splitting of water, due to pumping of protons from stroma to lumen, and due to consumption of protons from stroma by NADP+ to be reduced into NADPH plus H+, the proton concentration is decreased in stroma. So in stroma, proton concentration is decreased, whereas in lumen, proton concentration is increased. Concentration of protons is increased. Okay. So because of what? Because of 3 reasons, or because of 3 factors. What are those 3 factors? I am going to write those 3 factors here for the increase in the concentration of protons. So when there is increase in the concentration of protons or the number of protons present in lumen, what happens? Proton gradient is developed. That proton gradient is nothing but proton motive force. So now let's write the reasons or factors which are involved in the development of proton motive force or proton gradient inside the lumen of this thylakoid membrane. So here PMF, proton motive force, is developed. Proton motive force is developed. Okay, let's write the reasons or factors for the development of proton motive force inside the lumen. Okay, first reason is due to splitting of water. So the splitting of water is taking place towards lumen, and hence protons are released into lumen. This is one reason, one factor. Next reason is due to pumping of protons from stroma to lumen by proton pump. What is that proton pump here? PQ. So pumping of protons. Due to pumping of protons from stroma to lumen. From stroma to lumen. That is second reason, second factor. Third factor is due to the consumption of protons by NADP plus to be reduced into NADPH plus H plus, which is catalyzed by the enzyme NADP reductase. So this NADP reductase enzyme is involved in utilizing protons for the conversion of NADP plus into NADPH plus H plus. So NADP plus is converted into, that is reduced into NADPH plus H plus by consuming protons from where? From stroma. And hence due to these three reasons, what happens? Proton concentration is decreased in stroma but increased in lumen. Okay, resulting in the development of proton gradient. That is nothing but proton motive force. So now this proton motive force undergoes breakdown. Proton gradient undergoes breakdown, and hence proton starts moving towards stroma. From where? From transmembrane channel of ATP synthase enzyme. Now let's study ATP synthase enzyme first. What are the parts present in this ATP synthase enzyme, and how these protons are moving through this ATP synthase enzyme, and how ATP synthesis occurs, right? So this is ATP synthase enzyme, which is associated with the thylakoid membrane. This enzyme consists of two parts in it, two parts, that is F0 and F1. So this part is called as F0 part or F0 particle, and this part is called as F1 particle. So this F0 part is an intrinsic protein. Intrinsic protein because it is completely buried inside or present inside the thylakoid membrane. So it is intrinsic protein. F0 part is intrinsic protein. And what is the function of this F0 part? It is providing transmembrane channel for the transport of protons from lumen to towards stroma, that is towards F1 particle of this ATP synthase enzyme. So this is intrinsic protein. What is its function? Its function is to act as transmembrane channel. It is providing channel way for the transport of protons. Transmembrane channel for the transport of protons. Next, coming to F1 particle. So this F1 particle is protruding towards the outer side of the thylakoid membrane, that is towards stroma side, and hence it is called extrinsic protein. Extrinsic protein, and along with this it consists of catalytic activity. It consists of catalytic activity of this ATP synthase enzyme, which is required for the formation of ATP from ADP and inorganic phosphate. So this is having catalytic activity which is required for the synthesis of ATP from ADP and inorganic phosphate, right? Okay, now these protons, they are transported from lumen to towards F1 particle of this ATP synthase enzyme. So once these protons enter into the transmembrane channel and then into the F1 particle, the F1 particle gets activated to start synthesizing ATP from ADP and inorganic phosphate. So for every 2 protons that enter into F1 particle, there is synthesis of 1 ATP. I am writing here, there is synthesis of 1 ATP from ADP and inorganic phosphate. 1 ATP. So for the synthesis of 1 ATP, how many protons are required? 2 protons are required. So this is the process of synthesis of ATP, in which proton motive force is involved in the is responsible for the formation of ATP. Right. Okay. Now I am going to give you calculations that how by splitting of one water molecule, how many NADPH will be formed and how many ATPs will be formed, and quantum requirement, quantum yield, all these terms let's study now. So before studying all these calculations regarding ATP formation, how many ATP are formed, how many molecules of NADPH is formed for the splitting of one molecule of water, how many protons, how many electrons and how many oxygens are involved, all this. Before going to study all these, I just want to give a quick recap of this chemiosmotic hypothesis. So chemiosmotic hypothesis is given by Michel, hence it is also called as Michel's hypothesis. He gave this hypothesis to explain the formation of ATP during photophosphorylation, which occurs inside the chloroplast, and during oxidative phosphorylation, which occurs inside the inside the mitochondrion. Okay. So inside the chloroplast, where this proton motive force is developed, actually this ATP synthesis is linked with the development of proton gradient or proton motive force. So where this proton motive force is developed inside the chloroplast means it is developed inside the lumen, whereas in case of mitochondrion it is developed inside the intermembrane space. So how it is developed? During the transport of electrons or protons are pumped from stroma to lumen, and hence inside the lumen there is increase in the concentration of protons, that is nothing but development of proton gradient, that is nothing but development of proton motive force, which is responsible for the formation of ATP. So what are the three reasons for the development of this proton motive force inside the lumen of thylakoid membrane of chloroplast? Three reasons are, first thing is water splitting occurs towards lumen. So first thing, water splitting occurs towards lumen and hence protons are left into lumen. Second reason, due to splitting during electron transport, the proton pumping occurs from stroma to lumen and hence protons are accumulated inside the lumen, which are pumped from stroma to lumen. That is second reason. Third reason is, when the electrons are finally reaching NADP+, this NADP+, after receiving 2 electrons, it consumes 2 protons from stroma and it is reduced into NADPH plus H+, and this reaction is catalyzed by the enzyme NADP reductase. And hence in stroma, because of these three reasons or these three factors, inside the stroma there is decrease in the number of protons and there is increase in the number of protons inside the lumen. Okay, so that is nothing but development of proton gradient or proton motive force, which is responsible for the formation of ATP. So after development of proton gradient, these protons start moving towards the stroma through transmembrane channel present inside the ATP synthase enzyme which is associated with this thylakoid membrane. So this ATP synthase enzyme consists of two parts. They are F0 and F1 parts. F1 particle. F0 particle is present inside the membrane. So this is intrinsic protein and it is providing transmembrane channel. It is providing pathway channel for the transport of protons from lumen to towards stroma. That is towards F1 particle, which is facing towards stroma. Right. So here, what is happening when two protons enter into F1 particle, there is synthesis of one molecule of ATP from ADP and inorganic phosphate. How it is synthesized? In the presence of sunlight. Hence it is called as photophosphorylation. Phosphorylation is synthesis of ATP from ADP and inorganic phosphate. In the presence of sunlight is photophosphorylation. By the energy coming from the oxidation of substrate means oxidative phosphorylation. Here it is photophosphorylation. So this is the development or formation of, this is about the development of proton motive force, which is responsible for the formation of ATP inside the chloroplast, that is during photophosphorylation. Okay, now let's start with some numericals related to the number of ATPs formed, number of NADPH formed per splitting of one water molecule or two water molecules, and how many oxygens are released. And let's study two more terms, quantum yield and quantum requirement. Okay. So before starting with quantum requirement, quantum yield and other calculations of ATP, NADPH produced, I would like to discuss few more points here. That is, chemiosmosis requires chemiosmosis, this process of chemiosmotic hypothesis. Chemiosmosis requires membrane. It requires membrane. So what is this membrane here? Thylakoid membrane in case of chloroplast. What is the membrane in case of mitochondrion? Inner mitochondrial membrane. Okay, it requires membrane. Then it requires proton pump. What is proton pump in chloroplast? That is plastoquinone. Whereas in case of mitochondrion, what is proton pump? Ubiquinone. Okay, and it requires proton gradient. Proton gradient or proton motive force. Proton gradient, which is also called as proton motive force. So where this proton motive force is developed inside the chloroplast? In lumen. Whereas in case of mitochondrion, in intermembrane space. So where it is developed? Inside the lumen. Next, it also requires ATP synthase enzyme, which is required for the synthesis of ATP from ADP and inorganic phosphate. Towards stroma, it is involved in the synthesis of ATP in case of chloroplast. So in case of chloroplast, ATP synthesis occurs towards stroma. ATP is released into stroma. Whereas in case of mitochondrion, ATP is released into matrix. So this is all about chemiosmotic hypothesis. In the next video, I will be explaining about quantum requirement, quantum yield, and NADPH formation. How many NADPH are formed? ATPs are formed per splitting of one molecule of water or two molecules of water. So this is, I hope you understood all these concepts. Thank you, students.